much. We've just got a last section before we release you. And so I'm absolutely delighted to introduce the final two speakers for today. Now, our first speaker, I believe, is online. And is he here? Yep, fabulous, wonderful. So we heard today how important it was, if you're thinking about getting ready for clinical trials, is to understand how a genetic condition is affecting a range of individuals, and particularly how we do that accurately over time. So we were, CureCLCN4 has partnered with the Simons Foundation, and this is really exciting for us because this gives us a position to get our families to have this longitudinal data collection. We're delighted to hear an update from Dr. Paul Wang, who's Deputy Director for Clinical Research at the Simons Foundation, who will be hopefully giving us an update on our CLCN4 families. Thank you very much.
I'll keep going. I didn't hear anything, but I'll keep going. I appreciate that it's late in the afternoon for you there, and I can assure you that this talk will be at least somewhat lighter than the intense biochemistry and cell biology that you've just been hearing about. And what I want to do is to give you an overview of the Simons Searchlight project, how it works, and then to share information about its progress specifically for research on CLCN4. The data that we have in it, the stem cells that we are in the process of creating for use by the broader scientific community. Just a quick acknowledgement that the Searchlight team is actually quite large, and I actually contribute very little to the work of the team. I just happen to be available to speak to all of you today. We have another meeting going on in our offices in New York, where a large number of these people had prior commitments. I will highlight one person here. Simons Searchlight, Simons Spark, a lot of the clinical research that we do at the Simons Foundation is really led by and has been the brainchild of Wendy Chung, MD, PhD. She's a pediatric geneticist, and she has been leading this work for more than half a dozen years, maybe 10 years or so. Many of you may have heard that Wendy is actually departing from the Simons Foundation to go to Boston, where she will be Chief of Pediatrics at Boston Children's Hospital and Chair of the Department of Pediatrics at Harvard Medical School. The project will continue on seamlessly. It will continue to be funded by the Simons Foundation, although it will now officially be based at Boston Children's. Again, will be supported by the Simons Foundation as an outgoing grant to Boston Children's Hospital led by Wendy. So we don't foresee, honestly, any major changes in how the program will be conducted and its goals. Just to take a step back, for those of you who aren't familiar, the Simons Foundation is a nonprofit foundation based in New York City, which has broad interest in the basic sciences, including math and physics and the biological sciences. Within the Simons Foundation, there's an initiative called SAFARI, this Autism Research Initiative. And there are several programs within SAFARI, of which Searchlight, the second column here, is only one. Some of you may know us best for the fourth column there, funding through grants, primarily investigator-initiated proposals that go to researchers around the world, really, everywhere from Australia to the US, South America, Europe, the Mideast, etc. We also have created and curate a number of research resources. And some of those are listed in the last column. Animal model repositories, we've created a number of mouse and rat models as well. We have online informational resources, which we call SAFARI Gene and SAFARI Base. In the middle column, a brain bank, which we refer to as Autism BrainNet, for which we accept donations not only from people who have idiopathic autism, but also from people who have genetically identifiable causes related to their autism and developmental disabilities. In the first column, Spark is a very large cohort of individuals with clinical diagnoses of autism. And then the focus of today's talk, the second column, Simons Searchlight, which is a project focused on people who have genetic diagnoses regardless of whether or not they have an autism diagnosis. So even though the original motivations for everything in Safari was related to autism, we've come to understand that, of course, as you all well know, there is enormous overlap between autism and other neurodevelopmental disabilities. And in Simon's Searchlight, we are addressing those neurodevelopmental disabilities broadly for those who have a genetic diagnosis. We collect, as it's listed here, behavioral information, clinical data, and also blood samples. I'll talk about this more, and are in the process of generating iPSCs from those blood samples. This is a list, fairly current, I believe, of all the genes that we work on within the Simon Searchlight project. Our goal, listed on the left, is to be an international research program that accelerates science and ultimately improves the lives of people with rare genetic neurodevelopmental disorders, including both single gene diagnoses as well as CNVs listed in purple in this first column. This is an international effort. Our materials are available at this point in the languages that are listed on the right hand of the slide, with hopefully more to come, more in the works. CLCN4 identified there in the red box. Work, as I've mentioned, on a number of types of data, collecting the medical and behavioral histories, the blood samples. We synthesize this information and share it back with families. I appreciate that the people in the room today and online are primarily researchers, a few clinicians, and very few families, but part of the talk, that some of the slides that I'm going to show you are really addressed more to the families than to the rest of the people in the audience today. The data that we do collect is freely shared by application to qualified researchers, including both researchers in academia as well as those who are working in industry. And we have, in fact, shared data, not on CLCN4 yet, but on a number of other genetic conditions to interested and qualified industry researchers. We also have a platform to connect researchers with the families who have enrolled in Simon's Searchlight, really to facilitate further work by everybody for the benefit of the broader community. For families, we put together summaries of what we know about the specific genetic conditions that we cover, and also give them information about for example, the surveys, the rating scales, etc., that they have filled out and shared with us, and how their results compare to those of other individuals who have the same genetic condition. This slide is just intended to show you what some of the reports look like that we feed back to the family. On the right-hand side here, for example, this is not CLCN4. We show an individual's particular results here in the dark blue, and then what we see in the larger genetic community for that particular person in the light blue, on a couple of different scales. On the top, the Vineland daily living scales, and the second one is the Vineland socialization standard score. But we do this for a number of other scales as well. As I've mentioned already, individuals can donate a blood sample. This can all be done remotely. There's no need to go to any particular medical center or clinic to have this done. There are, of course, many commercial labs in the United States. One of the most common is known as Quest labs, and they can draw the blood and send it in to us. And then we are partnering actually with the New York Stem Cell Foundation, and this work is also partially funded by the Nancy Lurie Marks Foundation to create the induced pluripotent stem cells, which we then make available to interested researchers. Very briefly, for the families or others in the room who might not be familiar with iPSCs, you've heard about a number of different research approaches today, and I'm sorry I wasn't able to listen to all of the talks. Being based in the US, some of them are quite early. So I don't know if anyone is already working with iPSCs in the CLCN4 research community. I know that there have been mouse model talks, and of course, mouse rodent models have been invaluable in drug development, and I'm sure will continue to be very, very valuable. But ultimately, of course, mice are not humans, and we're always left with a question about how well findings in mice will translate to the human condition. With iPSCs, what we do is that we can start with a person who is affected by the genetic condition, and then from their from their blood, ultimately create these so-called stem cells, which can then be induced, well, they are induced to become stem cells. They can then be further coast to develop into more specialized cells, whether those are neurons, nerve cells, or liver cells, or really any number of other types of cells. So then really the researchers will be working with cells that are human, that have the particular genetic variant, genetic mutation that that patient had, and see what happens, see how those cells are different from cells that come from somebody who does not have the CLCN4 variant, and how they respond to various kinds of manipulations, for example, medical pharmacologic manipulations. So iPSC research is still relatively new, certainly compared to mouse research, but everybody feels that there's just an enormous promise in using them as a model for understanding what's happening in these conditions and what we might be able to do about it. Finally, I just want to, on a general level, highlight what we call our research match facility, in which we connect researchers directly with families, individuals who have the genetic conditions under question. I'll go through some of the standard data that we collect on everybody who participates, but sometimes for a particular genetic condition or for a particular researcher, there's deeper information that they want on some specific facet of the condition that we haven't covered yet in the surveys that we usually send out. And so we can reach back out to the families who are in Searchlight to collect that additional data. And we can also tell them about your other research projects. For example, if there is a project that requires an individual to come into the lab, to come into the clinic or something, we can inform the families through our research match platform, ask about their interests, and then potentially connect them, if they are interested, with the researchers who are running that research project. So what do we have so far on CLCN4 in Simon's Searchlight? Again, this is what it looks like for families who are participating. First, they sign up online, and we've had 40 people in the CLCN4 community sign up online. 25 of them have already provided a genetic lab report so that we can actually confirm their condition and know that they do in fact have some CLCN4 variant. 13 so far have shared at least parts of the medical history of the person who is affected. 16 have filled out at least some of the surveys which we provide to everybody in Searchlight. 5 have provided blood samples so far. And then this is ongoing. As you just heard Dr. Palmer mention in her introduction, it is a longitudinal project. So we don't just connect with an individual one time. We come back to them typically every year to get an update on their medical condition and their developmental and behavioral status. The information that we collect, these are the standard forms that we send out, as I mentioned, starting with the genetic lab results. We have various forms of medical history, other diagnoses, medications that people are taking, and then quite a number of standardized survey measures that are frequently used in clinical practice. Everything from sleep to the child behavior checklist, which is a standard measure of behavioral conditions, behavioral differences, social responsiveness scale, frequently used in autism, similarly social communication questionnaire, quality of life measures, seizure history, et cetera, et cetera. We're always reconsidering what we're doing to make sure we're using the best, most valid, and reproducible measures, and trying to cover bases that we haven't covered in the past. So there are frequently updates to the list of surveys that we actually administer. Coming this year, there's a new measure which was actually originated in the Angelman research community, so-called ORCA, Observer Reported Communication Ability, which is more appropriate for individuals with lower levels of communication skills than in comparison to the other surveys that we have been administering. Also something, for example, called the Pediatric Evaluation of Disability Inventory, which gives a more fine-grained but still reliable estimate of fine motor and gross motor abilities. This is something else that we're working on also. Not sure exactly when we will be able to deploy this, is collection of raw EEG data. So this is what we want to do here, is to collect the raw data from clinical EEG. So children who have gone to their local medical institution, had an EEG. Almost always those EEGs are stored by the local medical institution, and then with the permission, with the consent of the family, we'll bring those EEG data in and have an archive of multiple individuals, hopefully, with CLCN4 or others, of their EEGs so that researchers can access them easily rather than having to go to each individual medical institution. Just a quick visual, the families who participate in Searchlight see a dashboard like this that shows them the various steps in the process. Have they registered? Who has consented? Parents, the individual themselves, if they're of consenting age. Has the lab report been reviewed, et cetera, and then the various tasks that are available to them. We do also recognize that we are asking them for an important part of their day, for a significant amount of their time and energy, and so we do provide compensation in the form of Amazon gift cards. I don't know if we do that around the world with this Amazon, but it's something else for people perhaps in parts of the world that Amazon doesn't reach, if there are such parts of the world. But I know that we do something for everybody. So here's some of the data that we have so far. I think you had a talk early on about the genetic landscape, if you will, of CLCN4. And here's summary data. I'm not sure this is fully current. It's only on 19 individuals, showing that, of course, not everybody has the same genetic variant, and these are just some of the ones that are known to be pathogenic among the individuals with CLCN4 who are participating in Simon's Searchlight. And from the blood samples that have been donated, I'm very happy to tell you that we have 4 iPSC lines that are in the works and will be available within the next year. Specifically, in the first line of this table, the ASN 362, that looks like it's about 6 to 9 months away. That's still relatively early in our pipeline. But then the last 3 lines, the 489, 555, and 718, are further along in the process, and we anticipate that these iPSCs will be available in a 4 to 6 month time frame. Again, these are available to both industry and academic and other nonprofit researchers worldwide at a nominal cost of $120 per sample, and you can request them at any time by going to our website, which we call Safari Base, our database of both simple data and also other kinds of resources that are available. I think the website is base.safari.org, but if I'm wrong about that, you can easily Google Safari Base, and I'm sure you'll go right to that website. So again, hopefully in less than half a year, we'll have the bottom 3 iPSC lines available, and then within 9 months or so, the top line as well. I'll just briefly go over some of the medical history data that we've collected. Unfortunately, we don't have these data on a large number of participants yet. We really do welcome all families who want to participate, and we do try to make the experience as painless as possible, while recognizing, of course, that it does take real time to sit down at your computer and share all the data that we are asking for. As we have been talking about, there's quite a large number of variants that have been identified in CLCN4. There are gain of function variants, there are loss of function variants, and we have not yet parsed these data according to all of those different subgroups. But just to give you an idea of what we have, among those 7 individuals, we have 3 who are 0 to 4 years old, we have 2 in the early school age range, and then 1 each in the early teen years, and then an adult as well, who's 35, in the 35 to 39 year age range. So those are the people who have shared data with us so far. Not surprisingly, I'm sure 100% of them have reported some language delay or impairment, intellectual disability in 6 out of 7, autism in about half, and anxiety and ADHD diagnoses in 1 individual each. These are, of course, very broad summary level data showing the broad range of symptoms that have been associated with CLCN4 variants. I've heard a number of speakers talk about hypotonia. That's in the tallest, or the longest yellow bar in the middle. A couple of subjects also reporting that they have increased muscle tone. 1 individual with cortical visual impairment, 1 with cerebral palsy. I apologize, I don't know why there's spastic CP listed at the top. and CP lists at the bottom. But anybody who is requesting this information would be able to drill down and be able to tell, for example, if that's actually the same individual or not. Just from clinical visual assessments, 2 individuals with strabismus, 1 hyperophia, 1 astigmatism, and then additional medical issues as well. 1 with seizures, 5 out of 7 with some sort of neurologic condition. And you can see some of the other categories that we have listed on here. Again, obviously this information would be so much more valuable if we had more individuals participating. We hope that'll be the case in the coming months and years. And then we'll be able to understand more, obviously, about genotype-phenotype relationships and understand that hopefully in a very fine-grained level with specific genetic variants and their association with various clinical symptomatology. We ask about medication use. But just as an overall summary, of course, this is no surprise to the clinicians who are experienced with CLCN4. We are seeing intellectual disability, language delays and impairments, autism in the subset, low muscle tone in the subset, and vision problems with some frequency as well. So that is actually my last slide. Thank you for your interest. We really do share the goal with all of you who are listening today that we'll be able to facilitate research on CLCN4 ultimately for the benefit of the CLCN4 community.